Hi guys! So we've been doing crosses for heredity in the past couple days, and this video is going to look at some special types of inheritance and special types of crosses to be familiar with. The first is blood types. And there's a couple things going on with blood types. First of all, there's codominance. So we've talked about codominance before, where there's two alleles that are both dominant. But then in blood types, it's not just those two alleles. There's actually a third allele, and that third allele is recessive. So there are two dominant alleles, one recessive allele. And the way that we show the dominant alleles are with capital I's and then with a superscript, either A or B. So blood type A and blood type B, those are codominant, whereas blood type O is recessive, and the way we show blood type O is with a little I. So capital I with an A is dominant, capital I with a B is dominant, lowercase I is recessive. And so over here, this table shows you the different genotypes for the phenotypes that are the blood types. So the phenotype, remember, is a physical characteristic. So you can either have type A blood, type B blood, type AB blood, or type O blood. Type O is the recessive. So type O, the only genotype for that is little I, two copies of the recessive allele. I'm going to go back up to the top. For blood type A, there's two different genotypes you can have, because you can be homozygous dominant for blood type A, so having two of those dominant A alleles, or you can be heterozygous for blood type A, having one copy of the dominant A allele and one copy of the recessive O allele. For blood type B, you could have two copies of the dominant, homozygous dominant, or one copy of the dominant, one of the recessive. Then since we have codominance in the blood types, you can have two dominant alleles, but the two different dominant alleles, and that gives you type AB. So those are all the different possible genotypes and phenotypes of blood types. And then what does that mean for the actual blood? What do you see in the blood cells? Well, the A's, B's, and O's just refer to these surface proteins, or little like marker proteins that are on the surface of the blood cells. And so type A blood has these little A proteins on the surface. Type B blood has these little B proteins on the surface. Type AB blood has both of them, and type O blood doesn't have any of them. And this we're going to look at tomorrow in class to talk about how these proteins have to do with what blood types can give to other blood types. So we'll get to that in class. But first here I want to see how you do a Punnett square cross with the blood types. So you set it up just like you would any other Punnett square. The only thing that's a little bit different is that you have the superscript. So the big I just shows you whether you're dealing with a dominant allele, and then the little I shows you that you have the recessive allele. The superscript, the B or the A, shows you which dominant allele you're dealing with. So in this cross, we have an individual that's heterozygous for B blood, so the dominant allele for B, and then a copy of the recessive allele. So heterozygous for B, and then the genotype for this individual is AB, so one of each of the dominant alleles. So then when you cross them, you just combine the letters like you're used to, and you end up with what kind of blood types do you see here in this cross that they end up with? You have 25% AB blood, or a 25% chance of getting AB blood from these offspring, 25% chance, or 1 out of 4 chance, of having a homozygous dominant type B blood, 25% chance, or 1 out of 4 chance, in having heterozygous type B blood, and then 25% chance, or 1 out of 4 chance, in having heterozygous type A blood. So now if you think of the phenotypes that are going to result from this cross, you're going to have 50% chance of type B blood, 25% chance of AB, 25% chance of A. This table over here just lists all the possible genotypes again and what blood type they correspond to. Another different kind of case for crosses that we want to be familiar with are sex-linked traits. And that means that they're traits that are located on the X chromosome. So remember, we have 23, we as humans have 23 pairs of chromosomes, and that 23rd pair is the sex chromosome. So for females, it's 2 X chromosomes. For males, it's the X and Y chromosome. And so there's some traits that are on the X chromosome, and because of that, females have 2 copies because they have 2 X chromosomes. And so since they have 2 copies, they can be carriers of sex-linked traits. Sex-linked traits are the ones that are located on the X chromosome. So when we see carrier, that just means being heterozygous. So if there's some trait that's a recessive trait and a female is heterozygous for it, she's carrying an allele for that trait but not showing that trait. Whereas males, since they're XY, if there's a gene located on the X chromosome, they only have 1 copy of it. So they can't be carriers of these sex-linked traits. They either have the trait or don't have the trait. So they're either dominant or recessive. They can't be heterozygous. They can't be carriers. Some examples of these sex-linked traits are colorblindness and hemophilia. And there's some groups doing hemophilia for their project, so we'll learn much more about that coming up. But basically, it's a blood clotting disorder. So this slide here shows a cross for colorblindness, which is one of our sex-linked traits. And what you do is to show the cross, you use the X's and the Y's to show if it's male or female, and then the allele is a superscript on the X's, because remember that trait's only going to be on the X's. So in this cross, we have a mom, so that's a female. She's going to be XX, and she's a carrier or heterozygous, so 1 big C, 1 little c. Cross that with the dad here, the father, the male, that is XY, and so there's only an allele for this trait on the X chromosome, and he's dominant for it, so there's big C. So in this case, colorblind is the recessive trait. So the dad has normal vision. He has 1 copy of the dominant allele. The mom's a carrier. She has 1 copy of that recessive colorblindness allele, 1 copy of the dominant normal vision allele. And so in the cross, again, you set it up like a normal Punnett square, except you have the C's are the superscript, or like the exponent on the X's. And you combine the squares just like you did before. You're going to end up with 2 squares that are XX, so those are the potential daughters, and then 2 squares that are XY. And you see that when you look at the C's that are on the X's, each of the daughters is going to have 2 copies of C's. One of the potential outcomes for a daughter is being homozygous dominant, so having normal vision and not being a carrier, not being heterozygous. And the other potential outcome is a daughter that is a carrier, is heterozygous, having 1 copy of that recessive allele. So she's considered a carrier. Remember, males can't be carriers because they only have 1 copy of the allele. So for the 2 potential outcomes for the sons of this cross, you have one of the potential outcomes being dominant, having normal vision, one of them being recessive, and that would be a colorblind son. So these are the 4 potential outcomes of a cross between these 2 individuals for the sex-linked trait colorblindness. We'll have an opportunity in class to practice some more of these. Make sure that you're clear on what the inheritance patterns are for blood typing and for sex-linked traits, and make sure you're clear on how to do the crosses. I'll see you tomorrow. Have a good night.